Today I'll be talking about intratumor heterogeneity through the lens of gene regulatory networks, or more specifically, we'll be looking at functional differences across subpopulations in cancer. So to start off with, well, cancer, we have all seen it, heard about it, we don't like it. It arises in multicellular organisms, and it's characterized by uncontrolled growth of cells. Now, this could be due to multiple genetic and epigenetic factors. Now, well, wherever there is uncontrolled growth of cells, we're going to see evolution of cells, and we could either see expansion of cells from the source normal cells, or we could see that these cells kind of evolve and give rise to different subpopulations. Now, why is this an issue in at least in the field of cancer? Because if you're coming up with some kind of therapy and we're trying to treat some specific population of cancer cells, if the population is giving rise to different subpopulations, then it's possible that the therapy could not actually affect the new subpopulation. So, well, these subpopulations are loosely defined as clones. And how do we really identify these clones? Well, we look at their genetic or DNA data. More specifically, we look at the copies of DNA that's present across these different cells. Across normal cells, you generally see a uniform copy of DNA across all of the cells. And when you look at tumor cells, you generally would find regions where there are maybe amplifications or deletions of specific genes or regions of the DNA. And that's how we kind of characterize or cluster these cells into common groups. So for example, we have clone 1 or a subpopulation 1, where we can see a large amplification of copies of the specific set of genes right here. Well, we also have this idea of gene regulatory networks, and for those who don't know, in a nutshell, it's basically it's a network that basically depicts regulatory interactions between genes. So if you have, let's say, one gene that's directed to another gene within the network, that basically means that this one gene is somehow regulating the activity of the other gene. Now, this could be depicted as a simple structure, or it could be some kind of function where we have some weighted regulation between two specific genes. And this is really important generally in the cases of evolution, development, disease, to kind of understand and characterize functional differences. And with that, we can kind of see where we're actually going. So we have clones where they're distinguished on the basis of DNA copy numbers, but we haven't really looked into what exactly are the characteristics of these different clones. Can we really find functional differences across these different clones? So the point of today's talk would basically be just that. Can we utilize gene regulatory networks which help in characterizing functional differences and figure out these functional differences across clones? So to start off with some of the state-of-the-art GRN inference tools that we'll be using, CellOracle and GRN Boost. These are models which CellOracle is a linear model which prunes existing prior regulatory networks into something that's more data set specific. And GRN Boost is an ensemble method that basically tries to figure out the importance of a set of genes in regulating the expression values of another gene. And some of the existing approaches and some of the new approaches that we came up with to identify these functional differences would be node centrality measures, something that's really popular in network analysis that allows us to identify the influence of specific nodes or genes within a network, bottleneck genes. And we also have this idea of bootstrapping that we can see here, where we basically weigh out each of the edges within the network. Now, what does it mean to weigh out an edge with, let's say, a bootstrap support value of 10? That basically means that if we subsample this population of cells into multiple subsets, in this case 100 subsets, and we generate a GRN for each one of these subsets. A value of 10 basically means that this edge E1 is present in 10 of these generated GRNs. So the higher the bootstrap support value, that basically means that an edge is prevalent in most of the GRNs that have been generated from the subsampled GRNs. Yeah. Okay, with that, the first point would be, can we really see overall differences across clones and tumor through the lens of a GRN? So in this study, we considered multiple samples of triple-negative breast cancer and colorectal cancer, but for the sake of presentation, I'm just focusing on one specific example. So where we basically have triple-negative breast cancer with 2 clones, we perform GRN inference where we have clone-specific GRNs for clone 1 and clone 2, and we see large differences across tumor and normal, which kind of makes sense, but we also see a good amount of differences between the 2 clones. So which kind of hints at the idea that it already captures structural differences across clones and also has these differences between tumor and normal cells. But at the same time, we performed our bootstrap analysis, and what you can basically see here within this violent plot is basically the bootstrap values of the genes involved in these in the GRN. So what we have here are basically the unique edges. The orange section basically represent unique edges within a specific clone GRN, and the blue section basically represents all of the edges present within a specific clone GRN. And what's interesting to note is that the bootstrap values are slightly lesser for the unique edges, which will kind of make sense in some time, which we'll see later on. Now we can represent these bootstrap edges as binary vectors, where if you have an edge E1, so you'd basically have 1 if it's present in a specific bootstrap, and it would be 0 if it's not present in that specific bootstrap. So if you have 100 GRNs, if it's present in one GRN, it has a value of 1. If it's not present, it has a value of 0. That allows us to identify distances between intertumor and intratumor, so intertumor and intratumor distances. And what we could see is that we could see a larger intertumor difference in comparison to intratumor differences, which kind of hints at this idea that GRNs are able to capture global differences across clones and tumors. And further on, we kind of wanted to look into what can we really find if we apply network centrality measures. So if we had let's say if we had the clone-specific GRNs, clone 1 and clone 2, and applied some centrality measure, in this case we considered degree centrality, we noticed that a lot of the genes involved in these 2 GRNs actually had very similar degree centrality measures, but then we also had a couple of outlier genes. And what we knew, at least what we could find, was some of these genes that popped up were intensively involved in pathways related to progression of triple-negative breast cancer. And furthermore, we came up with this idea of looking at predecessor and successor Jacquard similarity. Now, what does that mean? Let's say that we have a gene of interest within the network. We have a set of predecessor edges and successor edges. We can compute a Jacquard similarity between 2 clones for a specific gene. It could either be Jacquard similarity on the basis of predecessor edges or successor edges. Now, what does that mean? If you compare the successor or predecessor edges between the 2 clones for the gene of interest, how different are they? If the Jacquard value is very low, it's a value between 0 and 1. If the value is really low, that basically means that there are a lot of differences. If the value is really high or close to 1, that basically means that there are very less differences between the 2 clones at that specific gene of interest. Now, what we did is, well, we just plotted a histogram of the Jacquard similarity values across all of the genes. And of course, it followed a normal-like distribution. where what we noticed was a lot of the outlier genes on the top, on the later quartile, basically represented housekeeping genes, which made sense because housekeeping genes kind of have very similar functioning across the two clones. But what we noticed was some of the interesting genes that fell at these lower quartiles right here, like for example HERPED1 and CDH19, which are known to be therapeutic targets or tumor suppressor genes respectively in triple negative breast cancer. Okay, so what did this actually help us understand? Well, we basically understood that GRNs can help us figure out overall differences across clone specific, overall functional differences across clones. But we wanted to see, can we find more biologically meaningful differences? So to do that, we looked into a study that considered time series data, where there are basically cells segregated into pre-treatment cancer cells, post-treatment 1 cancer cells, which are cells at a necrotic region of the tissue, and you have post-treatment 2 cancer cells, which are near tumor margins. And what we basically did initially was to compare the between centrality of each of the genes across the different time points. And what did we see? We saw multiple outliers that are again either involved in metastasis or are candidate biomarkers for tumorigenesis in the triple negative breast cancer sample that was used in the study. But what was interesting to note was when we looked at the clone specific or subpopulation specific differences across these 3 time points, we noticed that, well, there were some sort of irregular differences across the clones. So we looked at between a centrality measure and we saw that there were differences across the clones. But what was interesting was at the necrotic tissue, we saw that some of the clones were completely inactive and some of them were actually very inactive, which kind of again pointed out to this idea that clones actually have a very huge role to play in tumorigenesis. Furthermore, we wanted to look into, there was a study on glioblastoma and cell states of glioblastoma. So there were specifically four states, and the cells within the glioblastoma could be characterized into, well, it could be segregated into one of these 4 states. How do we define these states? Well, it's defined on the basis of meta-modules or a specific set of genes. So these set of genes are highly expressed or lowly expressed within a specific cell state or the other. What we see here in these plots right here are basically the distribution of these clones across the 4 different cell states. And what the authors mentioned was we don't really see clones belonging to a specific cell state, but we can see some sort of inclination of clones to one or the other cell state, depending on the sample that they were working with. In this case, we are looking at 2 specific samples out of 9. So what did we do? Well, we extracted the genes that belong to each one of these metamodules and created a subgraph from the GRNs. And we calculated a metamodule specific activity using a univariate linear model. Now, what you basically see here is the activity of this subgraph or metamodule across each one of the clones for each one of the cell states. One thing that helped for validation was, well, the trend of activity followed the trend that we can basically see in these 2 plots right here. But what's interesting is that the GRNs actually help us understand more fine-grained differences across these cell states, which is pretty neat. So overall, what we tried doing was we tried to see whether we could apply these set of analyses or tools that we built to understand functional differences across clones and characterize clones better. But what we think could be done, and something that we are what we are currently working on, is can we consider multi-model data sets so that we can get more accurate findings? Can we perform these analyses more efficiently, and are there more sophisticated approaches that will help us, characterize these differences a lot more better? So finally, I would like to thank my supervisor, Luai Nakle, and Professor Hamim Zafar, and Dr. Nikolai Shapoval who have been really helpful with their insights and contributions in shaping up this work. And also members of Nakle Lab for encouraging me throughout the way. We have a QR code for the preprint that's currently online. And with that, I'll end my talk. Thank you.
Thank you.